We'll start our tour of the eukaryotic organelles by first looking at the nucleus and the endomembrane system. The nucleus is where we start in this discussion, and the endomembrane system is really just an extension, physically anyway, of the membrane surrounding the nucleus. So these are all interconnected and make for a good set of organelles to discuss all at once. I have a picture of both a eukaryotic cell and the next, or both a plant animal cell and then next also a plant cell, and I'm pointing out this extension from the second outer membrane of the nucleus. This extension right here and also this one, these are called the endoplasmic reticulums, or the rough and smooth ER. Also, the Golgi apparatus is an extension, part of the endomembrane system, as well as the lysosomes. Lysosomes don't look to be physically connected, and neither does the Golgi, but we'll find that all these membranes are derived from the same place, and so they're all part of the endomembrane system. In the plant cell, same setup. We're going to see that the functions of these organelles are the same, but I want you to notice that what's missing in this picture is a lysosome. Lysosomes tend to not be found in plant cells. The things that happen in lysosomes tend to happen in the vacuole, which is part of the next lecture. So first, the nucleus. The nucleus is where DNA is housed, so it's where we have all of the copies of the genes or the sets of instructions for making proteins. It's well protected in there. It's a organelle that has a double membrane, so two phospholipid bilayers surrounding it, and we call that double membrane the nuclear envelope. Within the nucleus, the DNA is housed as chromosomes, and that copying of a gene into RNA or transcription occurs within the nucleus. The nucleus also has pores because things have to get in and out of the nucleus, and those pores are gated, so they have proteins that line these pores, and the proteins provide a gate to allow certain things in and certain things out of the nucleus. There are lots of proteins in here doing different jobs. Lots of proteins are involved in transcription, for example. There are structural proteins in here, so enzymes, structural proteins, cytoskeletal proteins, lots of different types of proteins that get made initially in the cytoplasm and have to come back in. And then things like mRNA have to be able to, or RNA have to be able to leave the nucleus in order to be copied into proteins. So there's a lot of movement in and out of the nucleus. Last, there's a spot in the nucleus called the nucleolus. It's not membrane bound, but it's a specific region within the nucleus, and this is the place where ribosomes are assembled. We're going to see that ribosomes are assembled in pieces, and those pieces are assembled in here, and then they're sent out of the nucleus in order to be able to then create proteins in the cytoplasm. Now, an extension of the outer membrane of the nucleus is the endoplasmic reticulum. The smooth ER is multi-purpose. It's the site primarily of lipid synthesis. So any kinds of phospholipids, triglycerides, pigments that are related to photosynthesis, all these things are synthesized in the smooth ER. These things are built by proteins, so they're not encoded in DNA directly. Instead, there are enzymes that build the different types of lipids from precursor molecules, and that all happens in the ER. In addition, there's some detoxification of different types of chemicals in the smooth ER, and the smooth ER is a site of calcium ion storage, which is a ion that the cell uses to transmit signals within the interior of the cell. The ER is, of course, membrane bound, and it's a single membrane, one phospholipid bilayer, and the interior of the smooth ER is called the lumen. There's another type of ER called the rough ER, and the rough ER is still single membrane, and it's an extension of the nuclear membrane, one phospholipid bilayer surrounding the lumen of the rough ER. Now, the job of the rough ER is to be a site of protein synthesis, but it's only one site of protein synthesis. RNA leaves the nucleus once it's been transcribed, and ribosomes in their pieces assemble into one large unit on mRNA in the cytoplasm. And if a protein is one that is to be secreted, so sent out of the cell, or to be involved in the plasma membrane, so to be a protein that's actually spanning the membrane, or if it's one that acts within the endoplasmic and the endomembrane system, then the ribosome that's starting to translate it in the cytoplasm is going to move to the rough ER and finish synthesis of the protein into the lumen of the ER. So this is called a rough ER because all along the outside of the membrane of the ER you can find little ribosomes, and under the electron microscope they look like little bumps on the ER. And that's missing from the smooth ER. We don't have these little bumps because there's no protein synthesis and no ribosomes. So these little circles here, these bumps on the membrane, are the ribosomes, and you can't see RNA or the polypeptide being produced, but they are synthesizing proteins and pumping those proteins into the lumen of the ER. So the ribosomes are actually out in the cytoplasm of the cell. Once they get assembled into their pieces, they're sent out into the cytoplasm. mRNA is transcribed, and the mRNA is sent out into the cytoplasm, where the ribosome grabs hold of it and starts the translation, starts to synthesize the protein. These ribosomes translate nucleotide language into amino acid language. They read the sequence of nucleotides, which is several thousand bases long in RNA, excuse me, and translate it into the sequence of amino acids. And like I've said before, that gene expression is part of a later topic. So this is actually a figure from your text. I'm kind of mixing different text figures here. But outside of the nucleus, you're going to find a continuous extension of the nuclear membrane. Part of that is smooth ER, part of that is rough ER, and they have very distinct functions. Now, the Golgi apparatus is the next part of the endomembrane system that I want to talk about. And the Golgi apparatus sorts and sends proteins that have been synthesized by the rough ER. So here's our ribosome sitting on the membrane of the rough ER. And this protein that it's synthesizing is one that's going to need to be either secreted out of the cell in the plasma membrane or being sent into the endomembrane system somewhere to do its job. And that is determined right away when it first starts translating the protein. So that's determined by other proteins that aren't described here, but the ribosome travels over from the cytoplasm, binds to the membrane of the ER, and starts cranking that protein into the lumen. In the ER, the protein folds, and it gets chemically modified. Usually, if it's a protein that has sugars attached to it, because maybe it's a plasma membrane adhesion protein, and the sugars act as little glues, those sugar chains will get added in the rough ER. And once the ER has packaged up this protein and modified it properly, it actually sends it out by transport vesicle. This bud of membrane buds off of the rough ER membrane, and it is targeted to the Golgi apparatus. So this is a secreted or membrane-bound protein, and it's sitting here inside of the transport vesicle. Now, the Golgi apparatus is going to receive this vesicle. This membrane-bound vesicle fuses with the membrane of the Golgi. Remember, all this membrane is all phospholipid bilayer. So these hydrophobic bilayers fuse really easily with each other. And the Golgi transports this protein through its stacks, through the lumen of these different stacks of the Golgi, and within the Golgi, at the transphase, or the shipping side of the Golgi, these vesicles are formed again, and these transport vesicles are taking proteins away from the Golgi. The Golgi, during all this time, has determined where the different proteins go. Do they go to the membrane? Do they then stay there, or do they get shipped out of the cell? Or maybe the Golgi sends it back to the rough ER to go do some kind of job. Now, the Golgi is not attached to the ER, but the source of the membrane for the Golgi is the vesicles that come from the ER. So we still consider it part of the endomembrane system. All these membranes are derived from the same place, the outer membrane of the nucleus. Now, there's one more organelle that's part of the endomembrane system, and that's the lysosome. The lysosome is the site of intracellular digestion. Inside of the lumen of the lysosome, there's a very low pH, and there are enzymes that are only active at low pH. Their job is to break down macromolecules. And it's really good that these enzymes only work at low pH, because if the lysosome was to rupture and these enzymes were to go out in the more neutral pH, we wouldn't want them to start degrading all of our cells' macromolecules. So inside of here are these enzymes, low pH, where any kind of macromolecule that needs to be broken down can be broken down inside of the lysosome because of the intralysosomal conditions. These enzymes for the lysosome actually come from the Golgi. The ER has made the protein, the ER sent it to the Golgi. One of the many transport vesicles coming from the Golgi could be carrying digestive enzymes to the lysosome, in addition to the different types of proteins that go to the membrane. So this vesicle is coming from the Golgi apparatus, full of enzymes to digest macromolecules. One source of the macromolecules could be an ingestion of food particles, things that haven't completely broken down into their various pieces by the digestive system, can be taken in by cells, and this membrane-bound vesicle coming in by endocytosis can fuse with the lysosome, and then these enzymes that have been activated in the lysosome will break down the food into small components that can be used by the cell. This is also what happens like when white blood cells ingest bacterial cells. They destroy them for you, and the endocytosis results in a vesicle that's carrying the bacterial cell, and the lysosome will break that down and destroy it for us. The lysosome can also be used to break down worn-out organelles, because our organelles aren't permanent. They're always getting made and recycled, made again. The lysosome can fuse with an internal vesicle carrying a damaged organelle. Again, they fuse with each other. The lysosome breaks it down into component pieces that can be reused by the cell. So if we take this all together, we've got the nucleus and the ER. We've got the Golgi and we've got lysosomes. The nucleus is constantly going through transcription, gene expression that also includes translation, in order to create proteins that the cell needs. Maybe those cells need to go out of the, maybe those proteins need to go out of the cell. Maybe those proteins need to be embedded in the membrane. Or maybe those proteins need to go to the lysosome or back to one of the ERs. If there are any of these types of proteins called secretory proteins, they have to go through this process, ER to Golgi, and then transported out. Remember that the Golgi is sending vesicles to different places. And what's really cool about the Golgi is that it identifies proteins based on their amino acid sequence for targeting to the correct spot. Now, there are other proteins that the cell makes that don't go through the secretory pathway or through the endomembrane system. Like some proteins have to go to the nucleus. Some proteins have to go to other organelles we haven't discussed yet, like the mitochondria and the chloroplast. Ribosomes don't always travel to the rough ER to finish protein synthesis. Sometimes that's just completed in the cytoplasm, and the proteins that are made then go to the right spot. But the endomembrane system is the place where we get the synthesis and then targeting of special types of proteins that I've discussed as the secretory proteins. Now, the next lecture video is going to discuss some of those other types of organelles and their function, those not related to the endomembrane system.